Bi- Bien, bonjour, bonjour à toutes et tous. Bon, aujourd'hui, on a le plaisir d'accueillir Tullio Reverso qui a fait donc qui a fait ses études en Italie, hein, et puis ensuite sa thèse au LEDIX. Je pense qu'il va nous parler de ce sujet-là. Puis il a continué ensuite à Manchester ou à l'Alan Turing Institute sur la modélisation de la turbulence. Et pour l'Imperial College, je ne me souviens plus. Ah, d'accord, ok. Et donc, il est actuellement donc à Saint-Gobain. Ben, à toi, Tullio.
Aujourd'hui, je vais vous parler de mon projet, une partie de mon projet de thèse que s'insère dans le plus grand après projet que j'espère de finaliser bientôt pour le concours CNRS et que est basé en partie, en grosse partie, est basé sur justement mon travail de thèse et tout ce que j'ai pu comprendre qui m'a un peu inspiré. Si ça vous dérange pas, je parlerai en anglais seulement parce que c'est un sujet que j'ai jamais présenté en français. J'ai un peu peur d'être que ça va prendre beaucoup plus du temps si je parle en français. Donc, we're going to look at the problem of confined active suspension. So that's a fluid that it per se behaves like a Stokes fluid, so very low Reynolds, with some particles in it that are forcing this fluid. And so there's a coupling between the behavior of the suspension and the behavior of the fluid. We're going to see a little bit of motivation. I'm going to describe the system at the microscopic scale because we look at one particular swimmer. Then the good thing is these models are very widely applicable. So it's not really restrictive from a sufficiently far away perspective. Many biological and non-biological industrial fluids can be modeled at least in the same way. We're going to look at their collective behavior and the final part, which I think is the most important, especially for actual application, is the response of these fluids to active forcing and confinement. Just a few, I'm going to go through the why and what pretty fast. So this is an example of collective behavior. What I want you to notice is that there are at least two length scales that are involved. Actually, I would say even more, three at least, because we have the size of the bird in this case, the size of the flock. Their difference is very large. The ratio is very small, depending how you put it. And the important thing is that there is a third, actually, length scale. That is the length between the interactions of the particle. That's very important. It's pretty small, actually. These birds probably interact just with their neighbors. They don't really look very far away. And but the correlation of their motion, so the long length scale, it's much larger. So it's typical in a problem with many interacting particles. So the information propagates, and even if you have a small interaction range, you can have collective motions at much larger scales. An example from the microscopic universe is this E. coli, very well studied already in the 90s. There were some, even before, there were some experimental study. What are we looking at? So we're looking at suspensions of those little guys that have a self-propulsion capability, so they swim, and they interact via chemical interaction. So basically, they produce a chemical species that diffuses around them and attracts the other. And that's why, if put into this is a drop of hydrogel, they end up forming these little clusters. At first, you can imagine this as being uniform, and they are more or less evenly distributed. And after some time, they form these evenly spaced, maybe regularly spaced clusters. And that's chemical interactions. The same bacteria also produces this kind of motion if in a less viscous fluid. This is called bacterial turbulence sometimes, even though it's a little bit of an abuse of, it's not really turbulence. But this is another time the outcome of their interactions. This can be explained either via hydrodynamic interactions and probably to get actually very accurate representation, we should also consider the steric interaction between the bodies, in the sense that the contact, because here it's very dense. That's just another example, biological. Let's switch to stuff that it's not biological. So here are little particles that swim into their, basically they catalyze a reaction on their surface in an asymmetrical manner, and they manage to self-propel. This is a 2D kind of situation, so they are on a plane. And they form these clusters. What I want you to notice here is there are clusters and a gas phase in between. So my work will focus mainly on modeling the gas phase and how it can transition or not to cluster. The cluster itself, I won't be modeling because I use a continuum approach, so it's hard to take into account steric interactions in this kind of modeling. So this is an example of artificial stuff, let's say. All right, so let's look a little bit at these just to have an idea of where the model comes from. When you have a little particle like this submerged in a fluid, and you imagine the fluid, and in the fluid there is a solid, we'll call it C for now, and these are the blue dots. There are many ways, but one of the way in which a particle can propel is if there's a gradient of this solid. And depending on the nature of the solid, this can happen in different ways. The simplest, I think, is the diffusiophoretic mechanism. And it's basically it's driven by the fact that the blue particles and the white particles, which is the suspending fluid, have a different interaction potential with the surface of the particle, of the colloid, and this drives a tangential stress, and therefore to equilibrate this tangential stress, the particle moves in a given direction. So to cut it short, we have a relationship between a chemical gradient in the bulk and the slip velocity that emerges on the surface of a particle. And that depends on the property of the surface of the particle in relation with whatever is dissolved into the fluid. There is a coupling, hydrodynamic and chemical. So we need at least two fields to model all of this. Since we want to talk about swimmers that propel themselves without the need of an external gradient, we can think of swimmers like the one before. There is a part of the swimmers that drives a chemical reaction and produces this chemical gradient without the need for that being imposed, and it moves along a certain direction. So this is just a very, very simplified vision, but it allows to explain a lot of things. As I said, coupling between chemical and hydrodynamic problems. Very short slide here is just to say we're at 0 Reynolds, basically. It's enough to look at the order of magnitude of the quantities involved to notice that we can thankfully use linear equation to describe the motion of the fluid, which helps a lot because, well, mainly because, well, it's obvious why, because it's much simpler and we can superimpose effect. And for once, when we talk about fluid motion, we can think of superposition and of effects and not being totally wrong. Just another slide that is a little dense, but I'm going to cut through it because it's actually a lot of details that I probably don't matter very much. What I want you to retain from this slide is that we have said there is a link between chemical gradient and slip velocity. Now, the way these two are linked depends on the property of the surface. As we said, this link is through a proportionality constant called mobility. Depending on how this mobility is on the particle, you have all types of swimmers. So you have, especially you can have swimmers that are very stealth, that do not perturb the fluid in the sense that the slowest decaying perturbation relaxes as 1 over r cubed, and they're basically invisible at long distances. And there are some other that are puller or pusher, very much like the bacteria we saw before. There's the bacteria that swims like this and the bacteria that swims like this, and these are the same. In these, the case is 1 over r square, the hydrodynamic disturbance. 1 over r square is the same distance at which a chemical gradient decays as well, because these particles are a source of this yellow solute here. So this also leaves a trace in the far field. It's 1 over r, the concentration, because it's a net source. It's a point source. But the gradient is, which is what matters, the case is 1 over r squared. So we have a similar dependence, similar decay of chemical and hydrodynamic interactions. When we look at the far away, and we average the effect of all these particles in a unit of fluid, what happens is that we can write a tensor. There's this active stress tensor, and that works as a forcing, a local forcing, and unsurprisingly depends on the direction of the particle P and the gradient of the chemical field G. So that's all. We're going to look at some of these cases in details, and we're going to try to answer this question. So what causes collective dynamics? What is the role of self-propulsion, so whether we swim fast or slow, and what is the effect and the interplay between these two types of interactions that we're modeling. So how do we model a suspension already? Suspension, well, this equation can be derived from first principles, but of course we're not going to go into that. It's also not really my field of expertise. I mean, in the sense, we derive the coefficients from first principles, and those are very they can be derived analytically, the coefficients that I'm going to describe, but the equations themselves are it's a whole other story. In any case, it's not hard to understand that this is a conservation equation for a probability, and the way a probability, so the probability of finding a particle at point x directed along the vector p at time t, and the way it evolves is described by the motion of the particle. That's pretty intuitively correct. I think there's not a big surprise. The way the particle moves is via translation, so they swim in the direction p, they're advected by the fluid, and they move into a chemical gradient, plus they diffuse a little bit, because we're looking at a very small particle where thermal noise is still important, which becomes a diffusion of probability at long time scales. They also rotate with the fluid, with the chemical gradient, and they diffuse a little bit. This is just to give you more details on why this is described like this. It's a many body problem of interacting particle that is approximated in the so-called mean field approximation. That is, we look at the motion of one particle that, instead of interacting with all the other n-1 particles, interacts with an average field. Well, that's as much as I'm going to say. More details are either here, it's an article very recent that really does all the derivation, or my thesis. We did it a little before, but I was certainly not the first to do it. So we described the equation for this psi, which is the probability of particle. It's coupled with the fluid via this tensor. I'll show you exactly how. There are the active tensor depends on the particle orientation and the chemical gradient locally. And what is this chemical gradient? Well, we describe it with an advection diffusion equation. So we need 3 equations to describe all of this. Equation for the particle density, equation for the fluid, equation for the solid. How is the solid coupled with the particle distribution? Via source term proportional to the local density of particle. That's because we don't care about their orientation in the chemical interaction, since it's a point source. And anyway, so this is a set of nonlinearly coupled differential equations that we solve and we do stability analysis of. And we see what makes this stuff behave not trivially. That is why a suspension of this particle doesn't remain just a uniform distribution where nothing happens. We first do some stability analysis. Stability analysis, no need to say much about that. All I'm saying is that the base state is a uniform and isotropic distribution with a uniform field. The important thing, I guess, is to remind you a uniform and isotropic distribution in an infinite domain has 0 hydrodynamic field. Obviously, that's true because it's only true because this is a statistical description. And so we say on average, if the particles are not oriented anywhere specifically, and we can add the contribution to the hydrodynamic field of each particle, on average, all these hydrodynamic fields cancel out. So there's no hydrodynamic interactions in the base state. That's an important part, because everything that, well, no, that's an important part, and we'll see why after. The first instability we find is what happens when particles don't even swim, so it's a fully symmetric particle that's just a source of solute. And if we have a uniform distribution and we perturb it, means somewhere there's more particle emanate, so sort of this chemical substance, and another particle nearby tends to drift there. And there you see the feedback loop that makes the instability grow. If the particle swims, if the particle swims, this effect of swimming might stabilize the suspension because it has its own life. It goes in its own direction, so it can escape the attractive field. It can be understood like this. If we look at dispersion relation, this is the growth rate as a function of the wave number without self-propulsion, and this is as soon as you add self-propulsion. So self-propulsion really, and this is the zoom of that little area there. It tells you that you can stabilize everything, go at the growth rate below 0. But most importantly, what we see is that it changes even when it's unstable. The faster you go, the smaller the bigger aggregates is. This is the first unstable wave number, so it's the biggest unstable structure you can notice. And it becomes smaller and smaller as you increase the self-propulsion. And I was very happy to notice there's some experiment actually, in my opinion, proves it. And I think I'm going to try to convince you why. These are two different suspensions. In this case, self-propulsion is very slow and also decreases with time because of how the particle works. The kind of instability is the same. This cluster forms because particles are attracted chemically in the way that I've just described. What we notice is that in this case, particles, on the other hand, swim faster. When particles swim faster, the gas phase between the cluster is stable. Nothing changes. It remains more or less always at the same density. The size of the cluster does not increase. It's fixed by this kind of instability mechanism. On the other hand, here, basically the velocity of the swimmer tends to 0 eventually. And what you see is that the small clusters, which are equivalent to these and these and these, start to cluster together again. So basically, the gas phase, the particles are trapped into these clusters more effectively because of their slow swimming propulsion. So this is an example to show that. In fact, this is not just mental exercise, but I think we can see some evidence of this. Voilà, that's just what I just said. Another simple mechanism, again, we're exploring the different possible configurations so that they give something interesting. I won't cover them all, don't worry. This is basically it. Another one is, instead of drifting in the direction of your fellows, you turn and swim. Okay, fair enough. In this case, because of the link between hydrodynamic and chemical interaction, if you turn, you must also have a force dipole as a forcing signature on the fluid. Therefore, we can expect some interesting thing in terms of hydrodynamic interactions. However, hydrodynamic interactions do not occur in the linear regime in the sense they are second order. They're not because of the shape of the particles, which is symmetric, but we can do this numerically. So what we do, we solve the system of equation in 2D. This is a full spectral Fourier, with the orientation, the internal coordinates, also Fourier. And we look at the instability. We start with the formation of aggregates. So in the linear, at the beginning of the instability, these kind of things form, and this is purely chemical. It's basically what we saw in the video, in the experiment at the beginning with the little clusters. And the particles are pointing towards it. What happens after is that there is an effect of the hydrodynamic field. And that is because particles are not anymore randomly oriented. The chemical interactions has induced some form of orientational order locally, and this generates a non-zero field, even in a mean field description like this, and well, this is the outcome. So here we plot, we can see that this tells you how particles are aligned with the gradient of the chemicals, so how chemical interactions are working, and at some point when you have the average flow field increases, these things saturates. So there is a sort of hydrodynamic induced disorder, we could call it. But so, okay. Last kind of thing, just very quickly. If particles are repulsive, they also can do an instability. So if instead of turning towards their fellows, they turn away from them. And well, if also it's more complicated than this, but in general, what happens is that if you are repulsed, instead of generating aggregate, you generate a traveling band where you're basically continuously escaping in one direction from the particles that are next to you. And also because you turn, these kind of particles have an hydrodynamic signature. Anyway, long story short, let's look at the simulation. And this is the initial part of the instability where there are traveling bands that move in a specific direction, which is random, and they point away. Now, if we go on, we see that the effect of hydrodynamic interaction at some point kicks in, and the long-term dynamics are way less. But it's again the same effect as before. There's a sort of mixing. Again, I want to show you something that prove you that these kind of things actually happens, and it's not just a mental exercise. I found once in a conference, it was 2022, I think it was APS, a group from Princeton that did this very nice experiment. So what you see here is E. coli. So we said E. coli can form aggregates because they go towards each other, but they also can be anti-chemotactic in the sense they can also be repelled from each other. Because there's different strain of the microorganism, and in this case they are repelled by the fact that the other fellow's organism consume oxygen, and so well you want to stay away from the region where your friend has consumed all the oxygen, so they escape, and look what happens. They are exactly traveling waves that are observed. They are also in a gel, so hydrodynamic interaction are virtually zero. Everything is extremely viscous, but they are in a gel. They're put in this shape using I think a sort of 3D printer. And well, now, so just takeaway message: self-propulsion does have an effect quantitatively and qualitatively on the kind of instability we see. And well, chemical and hydrodynamic interaction matters both, especially in the longer term dynamics, not necessarily in the instability, the rays of the instability. Now let's see how these particles respond to external flow and confinement. I'm going to show you examples from the biological literature because that's what people have experimented the most in general, and they're more interesting thing. But curiously, I mean, the models are exactly the same. It's just a different interpretation of the various coefficients. So here we see the famous chlamydomonas in a Poiseuille flow. Maybe, yes, I think it's a Poiseuille flow. And we have that depending on which side we turn the light on, because these particles are attracted by light, the peculiar interaction with the combination, of the imposed shear, which imposes the rotation on the particle, and the direction from which the light comes on, can make them either focus on the center or on the sides. I didn't show the one on the side. You have to change the orientation of the light that you put in. So I think the light is either from here or from here. I don't remember in this specific case, but it's not too complicated to work around where it goes. If the flow is like this, particles are oriented on average a little bit like this. So I think here the light comes from here, I would say, because it makes them go towards the center. But it's along the axis of the flow. And here, well, here is the bacterial turbulence we saw at the very beginning, but if into a circuit becomes directional. So it doesn't, it really picks up a direction. They say race in this circuit. Another example, and this is a little bit just also for fun, but I find it very interesting, is how the dogs control the sheep. It is a little bit the same. The dogs are here, they go around and around and around. And their strategy, what I want to stress is that their strategy depends on the fact that this system has an internal intrinsic behavior. Like if each sheep was just ignoring each other, they should do a totally different job to get to the same point. So what I'm saying is if you want to control these kind of fluids or this kind of system, you need to have a simulation tool, a model that can reproduce to some extent their collective motion due to their interactions. You can just pick each sheep and bring them there. I mean, you could, but I guess this is more efficient. There's just 3 dogs for many sheep, so that works well. Voila, so let's see the simulation now, because when it's confined, this kind of model that I showed, as soon as you get confinement, analytical stuff and stability analysis by hand doesn't work because there's nonlinear boundary conditions and it's complicated. So we would just look at numerical simulation. We had to adapt the solver already, so we put everything into a channel, and we want to see the effect of imposed Poiseuille flow, and have a look at the effective viscosity of this channel flow, this fluid. So this is the thing we're using Chebyshev polynomials in this direction. We remain Fourier in the other, and we impose no flow. But already the particles tend to aggregate towards the wall. That's a very simple. It's an effect linked to self-propulsion. So the probability of finding particles becomes greater near boundaries, which is also observed experimentally in general. And voila, the effect is simply that particles that points towards the wall will go towards the wall and the other will go away. But when they get to the wall, they stay there, mainly as long as either their interaction with the wall or noise makes them go away. So in this case, the interaction with the wall does not make them go away, so you gotta wait for the noise. Voila, this is just what happens. So this is the stable, the base state of the situation. We got something like this, particle at the wall, pointing towards the wall, and not much more. If we wait, what happens is that they go and form. It's not very visible because the gradients are very big at the wall, so the color scale, I could have done a better job there. But we got 2 clusters, let's say, 2 big aggregate, not really cluster, 2 high concentration region here and here. Not surprising, it's happening exactly what was happening before. They form aggregates, except this time the aggregates are near the wall. Let's see what happens if we increase the flow. We got different dynamics. So this is the first one that I just described, formation of aggregates. The second one, we have a symmetry breaking mechanism top down. So we were very glad that we were not greedy and build a solver that is symmetric, because we didn't think there could be a symmetry breaking and we wouldn't have observed it. This will be much more boring without that, because the third one is fully symmetry breaking like this. And the last one, last one would have observed. It takes some time, and the colors, again, not maybe the best choice ever, but you should start to see some form of aggregates that travel. They are separated. So that's to say that with the interaction that I showed, the effect of confinement and of the forcing does have an effect, a qualitative effect on what we see at the end in terms of collective dynamics. Each of them has an explanation, as you can imagine, and we went through it, but I won't bore you with this now. Let's just have a look at the effective viscosity. So the effective viscosity, in this case, we used a very simple, basic definition of what the viscosity of a fluid is, which is non-local. In reality, is really based on the Poiseuille law. So I will describe this later, but it's all very simple. You got a viscosity, you extract the viscosity, you isolate the viscosity in the Poiseuille law, and you say, if for the same pressure gradient I get a different flow, well, I have a different effective viscosity. That's as simple as that. So what it means is that, I have forgotten to say one thing. In all the dynamics that you saw, the flow field generated by the particle didn't really have an effect, because we're very confined. There's probably an in-between between what we saw before and what we're seeing here. That is a spacing between the walls for which the hydrodynamic interactions play some role, but not now. But doesn't mean that the flow is zero. It doesn't mean that the forcing of the particles is zero. I'm talking about active forcing because, as you all know, when there is passive particles, there is a passive forcing. that is basically the fact the particles are inextensible, and so they increase the viscosity of the flow locally from a macroscopic perspective. Here we look at the active part. So that one always goes in one direction. If you put particle in a flow, the viscosity is going to increase, obviously. But if the particles are active, there is a second effect. And again, as I said, this is the luck of being in Stokes. You can actually add these effects together and figure out what happens. So this is just the flow by the particle. If there is a net, if these streamlines are open, means you are imposing a flow. If they're not open, you're not. Very quick takeaway message. I want you to focus on this plot and see that with time, the situation goes from the base state to some transient to the final state, where there are these two clusters that it's in a weak flow situation. And the blue line is a puller. No, it's a pusher. And the green line is a puller. So what you see is that, let's consider the pusher, for example. The pusher at first decreases the viscosity. What it means is that the flow field generated by the puller, by the pusher, goes in the same direction, is positive, as the Poiseuille flow. So you end up having more flow than without the particles. As soon as the situation changes because there are collective motions and self-organization and whatnot, this changes signs. So it means that, this is a very well-known result, by the way. So pusher swimmers are in general well known to be able to reduce the effective viscosity of a fluid. Here you see that if they aggregate, if they change their arrangement, that can change not only quantitatively, but even totally, in a sense, changing sign. So that was a takeaway of this. I don't have any experimental, any experimental proof that this is the case, but it can be engineered, and that's one of the things that I would like to do if I actually manage to get a research position somewhere, with some, well, by not necessarily by letting the thing evolve by itself, but by forcing the formation of this aggregate via external fields. That's the obvious first step experimentally, because you don't want to rely on too many things to happen. You just want to force some configuration of the particles and see whether it happens or not. And ideally here, if you have a phototactic swimmer, you put some light on the side and you measure the flow rate. Some experimentalists might tell me that's absolutely impossible nonsense. Some told me that it's possible. So it depends very much on how much you're basically able to measure, how precisely you can measure the flow field, and how easy it is to control those particles without, for example, killing them if they are algae, because they might. So I just don't want to take too much time. Where am I? Okay, 37. So I guess I'm going to spend very few minutes to show you what I would like to do next. Oh, so yeah, this is the final message, I mean, which is what I just said. If based on basically a little bit of this, of what I've learned, and also of what I've done during my other research experience, that just to give you an overview, well, this is what I just talked about. I worked on data simulation, that is adjoint methods for state and parameter estimation, on model order reduction, in the sense, well, we had the luck of having access to turbulent fluid data near a bluff body, and not that one, that's just because the picture is good. And we worked on a autoencoder that basically, well, we compared the typical POD decomposition of a turbulent flow with a decomposition based on nonlinear functions. That is an autoencoder. Specifically, it's called mode decomposing autoencoder, which honestly was very tough, and it takes. Today, I don't think there is any competitor to simple linear decompositions, because it's very expensive computationally and it's not really interpretable. There's a lot of issues, but we identified some promising direction for decomposing in general solutions of PDEs and whatever. It doesn't have to be a flow, and that I'll stop there. So that's what I also worked on, and I worked on surrogate modeling of drop size distribution. So these are basilisk simulation that we use as training data. We adapted the very useful pulsed jet example to a flat fan jet, in this case especially. And we changed 3 parameters, the Reynolds, the Weber number, and the spray angle. And we built a surrogate model of the drop size distribution that you obtained. Here is the example with different Weber. And this allowed me to basically go into the field of surrogate modeling. Surrogate modeling being you want to build you want to model a quantity of interest based on data that you have that you might get via numerical simulation, and how to make this as data efficient as possible, in the sense how can you reduce the number of simulation you need. One of the takeaway message was that I learned and that I worked on, because everything you see was coded from scratch. So even the Gaussian process regression model, I know it sounds stupid to code it from scratch since there are a lot of libraries, but we needed one that did things that were not available. For example, building curves based on integral and moments of the distribution. And so we did. So basically that's just to say a little bit of work was done also on my side for modeling from data coming from simulation, and trying to basically, for example, inform the next best simulation. Why I'm telling all of this, because if we go back to the results of this part that I just showed you, it reminded me immediately, it drove my attention to symbiotic system, because we tried to see what the effects of forcing an active fluid does on the collective motion, and this is exactly what symbiotic systems do. Very famous example is this squid, which recruits little bacteria from the ocean in a very noisy ambient with a lot of pathogens by using a combination of changing the viscosity, imposing a flow, and using chemical interaction to attract them, all combined in different stages until it gets infected exactly by the one kind of bacteria it wants. And also plants. The plants, this one is nicer. You got two roots with different shape, and the and the green are different bacteria that form colonies on them. And this process is called infection, which can be a positive thing and not just negative, happens in a fluid environment, depends on the flow that we impose if this is soilless agriculture, and on the interactions that are typically chemical between the plant roots and the different species of possibly pathogens. So the research questions that arise are, can we model this kind of situation where there is let's say the boundary of our domain represent, for example, a plant or the squid or whatever you want, and we want to model more different types of particles and see what control strategies allows you to select one rather than the other, or so what can be, for example, and work this to do optimization. So you want to optimize, I don't know, the flow that you imposed if it's this kind of agricultural system, or you want to maybe build a filter for particles. This is very interesting work that I just discovered yesterday, so don't ask me details about it, but it appears that they managed to sort blood particles. In the blood there are different kind of particles, and with applying a voltage they managed to build a device that filters out this stuff. Imagine putting this into that and having a thing that filters pathogens and keeps only the bacteria that are promoting the growth of the plant, something like that. So these is all things that I would like to explore, and can be done, of course, with the numerical approach on my side, and maybe with the experimental aid eventually. So this is just what I've just said. If we put in the simple channel that I showed before, and these are the roots that you want to optimize and study the effect of, depending with the crucial thing is with more than one species. So see the different thing. So I'll leave it with this, and I thank you for your attention. And if you have any questions, thank you.
Question. Paola, Paola.
Merci. Je sais pas si je fais tu avais l'air assez sceptique que là sur tes résultats d'auto-encodeur, effectivement, on a vu avec Thomas Noël qui est là que on est assez d'accord que c'est cher, mais peut-être que c'est aussi, comment dire, excessif pour certaines applications. Et par exemple, il existe des choses plus interprétables et plus simples, comme des extensions d'ordre élevé, des décompositions POD. Tu as des décompositions d'ordre supérieur. Et par exemple, c'est développé, je ne sais pas si tu as suivi le travail de Soledad Leclanch à Madrid, qui a un ERC là-dessus. Et alors après, tu as d'autres choses, c'est-à-dire que tu peux combiner également de la POD et des auto-encodeurs, de façon de profiter des deux. Et à ce moment-là, en gros, tu as la POD qui peut faire une partie du boulot, déjà trouver les bonnes directions. Et après, les effets non linéaires, tu peux considérer que c'est des effets d'ordre supérieur. Et probablement, tu arrives à les capturer avec des auto-encodeurs peu profonds et donc beaucoup plus agiles à entraîner. Donc, Non, non, c'était un commentaire. Et donc, est-ce que tu as regardé ça? Est-ce que...
Sur le so-called mode decomposing autoencoder proposé par Fukakata en Japon. Et ce n'était pas peut-être la meilleure, la plus efficace des décompositions des architectures. Et oui, je suis au courant, pas du groupe que tu as mentionné, mais en tout cas, par exemple, de l'association de POD et autoencoder, et surtout le travail fait à l'ENSAM, par exemple, où ils ont construit l'autoencoder qui impose un espace latent avec le plus bas le plus bas nombre de modes.
C'est aussi l'inverse, c'est-à-dire que la
Okay.
première couche, c'est
Ah,
POD,
ok.
et après, par exemple, il y a Alessandro Parent et
Ok, ah ok, très bien. Mais j'imagine, en fait, je pense que... Ok. Bah, je vais certainement le regarder et après peut-être je me note après les noms parce que sinon j'oublie tout. Et oui, c'est vrai. Moi, en tout cas, ce que je retiens de ce que je vois dans le sujet, c'est que en fait, il y a beaucoup d'architectures possibles et après ils sont très dépendants de l'application, dans le sens que selon l'objectif, parce qu'il ne faut pas oublier que en général, surtout avec des écoulements turbulents ou des choses très complexes, on fait de la décomposition pour un bout, c'est pas juste pour le bout de l'affaire. Et donc, je pense qu'après, voilà, la forme exacte de la structure de l'algorithme, du modèle que fait la décomposition, va dépendre beaucoup de ce qu'on veut faire. Pour moi, selon ce que j'ai compris, la chose plus promettante, c'est de faire de semi-supervised. Bah, ça dépend, pour l'optimisation en tout cas. Donc, ne pas seulement minimiser un reconstruction error, mais mettre des sorties, utiliser des représentations, une partie de la représentation latente de notre écoulement, par exemple, de notre donnée d'entrée, pour prédire des quantités et donc faire en sorte de, si on a un vecteur d'espace latent, utiliser une partie pour faire la reconstruction et une partie peut-être que se croise un peu pour faire pour faire de la prédiction d'une quantité. Donc on a la partie unsupervised et on a une partie... Et donc moi je compte développer, explorer, parce qu'après c'est un peu de l'exploration au début, ce type des architectures pour le bout d'optimiser quelque chose. En tout cas pour le bout d'extraire. Voilà, on a dit on veut décomposer quelque chose. Bah, moi, je voudrais idéalement avoir une architecture qui me permette de décomposer en composants en fonction de leur importance pour une quantité d'intérêt. Pas juste, comment dire, de façon totalement insupervisée, où on a peut-être quelque chose de très efficace au niveau de compression de l'information. Voilà. Ouais.
Combiner du non supervisé pour compresser et du supervisé
Voilà.
pour...
Exactement. Merci. 6. Tac, 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 tac. Peut-être il y a une façon plus vite de le faire. Voilà, on est presque là. Ah, vraiment, c'est là.
Oui, c'est là.
Ok.
Alors, là, je vais faire un commentaire, mais je vais faire un sondage d'abord. Qui peut deviner ce que je vais dire?
Moi, non.
C'est un détail, mais peut-être pas tant que ça quand même. C'est pas du tout un starling, l'oiseau que tu mets là.
Ah,
Donc, si dans la salle, tu as des gens qui sont intéressés par les oiseaux, ils vont être un peu choqués. C'est pas très bien de choquer les personnes parce que tu n'as pas mis le bon oiseau. Ça, c'était
peut-être, oui.
une hirondelle et ça n'a rien à voir avec les starlings. D'ailleurs, on ne fait pas du tout le vol de trucs.
Merci pour la remarque.
Donc, regarde bien starling, tu fais Wikipédia et tu trouveras un vrai
Oh,
starling et
oui.
les ornithologues dans la salle seront moins choqués.
Merci, merci. Mais maintenant
Désolé.
que tu as fait la remarque, je pense d'avoir déjà reçu cette remarque une fois et d'avoir totalement oublié parce que ça me revient. Ça me revient. Oui, c'est clair, c'est clair. Là, on est d'accord que ça, c'est des starlings, par contre. Ok. Ouais, ouais, il y a une incohérence. Merci. Merci.
Bon,
À la slide 16, s'il te plaît. On nous a montré un set de quadifs qui peut représenter les systèmes que tu voudrais décrire. Il y a plein de paramètres.
C'est là?
Oui, tout à fait. Non, non, c'est bon. On peut s'arrêter là. Et il y a plein de paramètres là-dedans qui vont piloter la dynamique de ton système, que ce soit les coefficients de diffusion, à la translation comme à la rotation.
Ouais.
Et, et toute la dynamique, donc tu as des nombres dimensionnés qui vont piloter la dynamique de tes phénomènes, les apparitions des choses. Est-ce que tu penses qu'avec du machine learning, on pourrait trouver un moyen de calibrer ces paramètres qui normalement sont un peu estimés expérimentalement, mais littéralement au doigt mouillé?
Oui.
Merci.
Alors, merci. Déjà, c'est une question très centrale parce que c'est un peu un problème de tous les modèles du monde. En fait, quand on veut être quantitatif, le nombre de paramètres a la tendance à augmenter de façon un peu incontrôlée. Et là, on en a déjà pas mal. En général, carrément, la meilleure des choses, c'est quand les paramètres peuvent être dérivés à partir de mesurés, on va dire avec des manips spécifiques. Donc là, c'est le cas simple où je sais pas, la viscosité de l'eau, on la mesure, après on la met dans notre code et si on sait que ça ne change pas trop, c'est un très grand avantage de le savoir a priori. Carrément, chaque modèle, même comme pour la viscosité de l'eau, on a des modèles pour l'estimer. Donc, chaque estimation de paramètres a lieu à travers un modèle. Donc, on pourrait effectivement utiliser ce modèle pour faire de l'inférence sur les paramètres. Et c'est quelque chose que je voudrais regarder. Et il y a toujours un risque, c'est-à-dire c'est un modèle très complexe qui est capable de reproduire des choses qu'on ne comprend même pas trop bien, qu'on ne sait pas trop. C'est toujours possible de trouver un set de paramètres que se colle bien à notre donnée, mais qui n'a rien à voir avec la réalité. Donc, ça ne nous permet pas après de faire de l'exploitation, d'extrapoler. Mais en général, c'est un des enjeux de ce que je voudrais faire aussi dans ma recherche, d'essayer justement de soit construire les modèles parce que exprès pour estimer le paramètre, donc en essayant de garder la sensibilité sur certains paramètres très bas, sur certains juste pour pouvoir les estimer. Mais oui, ça peut se faire, mais c'est un sujet très grand. Et moi, en tout cas, je compte de le faire en général, et ça c'est vraiment le niveau d'info que j'ai en ce moment en termes de stratégie, à travers des méthodes, on va dire pas intrusives ou black box. Donc, je ne vais pas, par exemple, faire le modèle adjoint de ce modèle et faire de la simulation de données. Tout ça, pas parce que je crois que ça ne peut pas marcher, mais parce que c'est trop, ça va prendre énormément de temps. Et après, si tu as besoin de changer de modèle, tu es un peu dans les choux, il faut refaire tout le travail du début. Donc, pour moi, c'est important de le faire avec des approches bayésiennes, par exemple, où on utilise le modèle de façon comme un générateur de données et on ne s'intéresse pas des détails de comment le solver est fait, de comment... Hein, et ça me permettra, par exemple, d'utiliser différents modèles différents solvers et de mélanger des informations par plusieurs sources. Donc ça, c'est au niveau vraiment général comment je pense de le faire. Donc des approches bayésiennes basées sur black box optimization. Parce qu'après, l'estimation de paramètres, c'est un problème d'optimisation qu'on espère de pouvoir régulariser bien pour ne pas tomber justement sur des nombres qui n'ont aucun sens.
Et pour ce qui concerne les instabilités de ta solution active à un milieu confiné à un canal, tu nous as indiqué que les champs hydrodynamiques ne sont pas très importants. Mais donc, est-il nécessaire de les simuler pour pouvoir observer ces instabilités? Ce sont des instabilités que pilotées par la chimie et l'hydro, elle est négligeable. Et donc, tu as besoin de la simuler, l'hydro, ou pas, pour avoir
Alors,
les obtenir? Merci.
oui, merci. C'est tout à fait une question raisonnable. Euh, là, on ne voit pas l'effet de l'hydrodynamique parce qu'on est très confiné. En général, on n'est pas assez confiné déjà. Et dans un système réel, où on a par exemple beaucoup moins de confinement, parce que là, si on va regarder après littéralement le nombre de paramètres, ce type de confinement n'est pas vraiment ce qu'on aurait dans un système vrai. Par exemple, s'il y a des racines des plantes, là, ça doit être beaucoup moins confiné. Donc en général, on doit la regarder. La réponse vite est ça. Il y a des cas particuliers où ce n'est pas important, mais par exemple, c'est nécessaire de la simuler et de la quantifier si on veut regarder la rhéologie, par exemple. La réponse du fluide à un gradient de pression imposé, tu ne peux pas le prédire sans faire ça. Ouais.
Ok, ben, merci encore.
Merci à vous. Merci.